So the next DNA manipulation technique that we're going to have a look at is something called CRISPR-Cas9. Okay, so CRISPR-Cas9 is something that we originally found in bacteria, and we've found that it's a really awesome way for us to edit an organism's genome when we need. So where has this CRISPR-Cas9 system come from? Because I feel like if we understand how the bacteria use it, it'll make a lot more sense as to how we use it. So this CRISPR-Cas9 system is naturally occurring in bacteria. So what bacteria have, they have something called the CRISPR array. So CRISPR is an acronym and it stands for Clustered Regulatory Interspace Short Palindromic Repeats, which is a big mouthful. But basically, they have these CRISPR arrays that have these protospaces, okay, which I'll show you in a second. So they've got these protospaces, and what they do is they wait and wait and wait until a virus or a bacteriophage, they're called, injects their viral DNA into the bacteria, which is a threat. Okay, so when the bacteria encounters this, what it's going to do is it's going to incorporate that viral DNA into these spacer regions. Okay, so a protospacer becomes a spacer when viral DNA is incorporated into it. So what this does is it kind of takes like a mugshot, all right? So it's like, okay, I've got your DNA, I know you're coming for me, and I'm ready for you next time. So what happens when it is invaded again? The bacteria transcribes this region, okay? Just like normal transcription, it transcribes this region and into something called guide RNA, okay, which we'll go through in a second. It transcribes this region into guide RNA, which attaches to Cas9. So Cas9 is an endonuclease, which we learned about in our last lesson. So it's going to go around and snip DNA. So how this works is this guide RNA attaches to this Cas9, it guides it to viral RNA, and when it encounters it, it will snip the viral DNA and destroy the virus. Okay, so that's how it acts as an immune mechanism in bacteria. So I'll draw it out so it makes a little bit more sense, and then we'll show you how we use it in the lab, which is very similar. So we've got our bacteria, okay? We've got our bacteria and we've got, that is terrible, I'm going to redraw. And we've got our CRISPR array. We've got our CRISPR array. Okay, so we've got our CRISPR array here, and it's got all of these protospaces. Okay, so proto just means like before, so it's before anything has happened to it. Okay, this is our bacteria. What's going to happen is a virus is going to come along, wants to infect our bacteria, and the virus, let's see how well I can draw a virus, not very well. I'll give him a mean face because he's mean. This is a virus here. Okay, this virus is going to inject his DNA into this CRISPR array. Okay, I'll CRISPR, and then it becomes a spacer, and these spacers have viral DNA. Okay, so CRISPR array gets injected with viral DNA and it becomes a spacer. So what happens next time that this virus comes to attack this bacteria again? So he's back, he wants revenge, he's back. Okay, what's going to happen is this spacer region that is already in the bacteria sitting there waiting is going to be transcribed. Okay, actually I'll put that here. So it's going to be transcribed into something we call guide RNA. Okay, guide RNA. So, and it's gRNA for short. So I might actually make this a different color. So this is our transcribed sequence with the viral DNA inside. Okay, so it's got viral DNA in there and it's called guide RNA. Okay, it's called this because it guides. All right, and we're going to see how it does that in a second. So this guide RNA is going to float off and attach to a Cas9 endonuclease. Okay, so we've got our Cas9 endonuclease here, Cas9, which is an endonuclease, and that will snip viral DNA. So what it's going to do is this guide mRNA is going to attach. Okay, so the important thing about this guide RNA, it's got this special sequence on it. It's got a special sequence on it called a PAM sequence. Okay, and this PAM sequence is going to allow the Cas9 to be able to recognize that it is viral DNA that it needs to attack. So it's not bacterial DNA because, you know, what's stopping Cas9 from attacking this? It's the PAM sequence. So the Cas9 will attach to guide RNA, recognize this PAM sequence, and then it's going to go around to all of the viruses and cut their DNA in specific spots. Okay. So when the DNA is cut, and I'll do this in a different color, so it's going to, where's my virus? I'll draw him over here. So he is being cut. Okay, so when it is cutting the viral DNA, what's actually happening is it's not the cut that is hurting the virus. It's not the cut. What's hurting the virus is the ligases that are coming over to repair it. Okay, so a ligase is going to come over and try to repair that DNA because that's naturally its job. It doesn't care whose DNA it is. It's going to try to repair the DNA. So while it's doing that, ligases, unfortunately, they're not very good at their job. So when they're repairing DNA, mutations happen often. Okay, so mutations always happen when ligases are repairing DNA. So it's hoped that the ligase will cause mutation. And it's the mutation that's actually going to render the viral DNA useless. And so it doesn't kill it, it doesn't snip it. What it does is it mutates it so that it is no longer functional. Okay, so let's have a look at that process summarized. So I've summarized it on the left-hand side. This is how it occurs in bacteria, and then we'll go through the right-hand side, how we utilize it in a lab. So just as I said, the viral DNA is injected into bacteria and is incorporated into a spacer region. So they're originally protospaces, they become spacer regions of the bacterial DNA. This DNA sequence is then transcribed into guide RNA and has those special PAM sequences, which basically help the Cas9 to not hurt bacterial DNA. These PAM sequences are what is recognized and attached to Cas9. So then we have the Cas9 guide RNA complex, and this endonuclease is going to cut DNA in those specific spots. So every time it sees viral DNA, it's going to cut it. It's not the cut that affects the virus. It is the ligase making its silly mistakes that is what we want. Okay, so the ligase is going to come and try and repair it. Not very good at their job, and it's hoped that mutation will occur to render the viral genes non-functional. Okay, so that's how they target viruses. So on the right-hand side is how we use this, and it's really similar, but obviously we're not doing this inside bacteria. We don't need to do that because we have technology. So what we do is we will isolate a DNA sequence for a gene of interest. So instead of using viral DNA, because we don't really care about viruses that attack bacteria, we might pick a cancer gene or some other gene that we're wanting to knock out or eliminate. Okay, so something that's going to be detrimental to our survival. So what we do is we isolate this gene and we make it, so in like a petri dish. Okay, we also make synthetic guide RNA for it. So we'll synthetically transcribe that DNA sequence of interest, and we make our own guide RNA that's called sgRNA. So that just stands for synthetic guide RNA, which is produced and then mixed with Cas9 in the petri dish. So then when the synthetic guide RNA and the Cas9 combine, they do the same thing that they would do in the bacteria. So the guide RNA is going to lead the Cas9 to the sequences that need to be cut. So in this case, it might be a cancer gene. And then we're utilizing the same mistakes of the DNA ligase to render the gene useless. So the ligases are going to come and they're going to try to automatically repair the DNA. So we are hoping for and betting on a mistake to occur, which is either going to help us to replace a faulty gene with new DNA, okay, or it's going to knock out or silence the target gene. Okay, so the ligase is probably going to cause a mutation in that DNA, which is going to silence the gene, and whatever, protein or damaging thing that gene was going to cause, can no longer cause it because it's mutated and can no longer function. So we still utilize those ligase mistakes. It's just this first part here that is synthetically made in like a petri dish, whereas over here it is inside of the bacteria and is organically made, and they are the two differences between those. So it's really important that you can understand both, how it's naturally occurring and how we're utilizing it in a lab to knock out genes that we don't want anymore. And it all has to do with those ligase mistakes. There is some things that we need to consider though with CRISPR. CRISPR is a very new technology, and often they'll like to ask you to think about the ethical considerations of this sort of technology. So because it's so new, we're not actually using it yet. So this is mostly animal studies that we've been looking at. So there's no human studies on this yet. So CRISPR cuts DNA at a chosen site, but scientists still have to introduce the nucleotide sequence of interest and hope it gets incorporated or knocked out. So we're still relying on that ligase making a mistake. And sometimes, like, ligases are pretty good at their job. They're not going to make mistakes all the time. So we're really relying on that mistake to be made, which makes it not very reliable as a technology. And also, to successfully alter an organism's genome, scientists must treat an embryo prior to the cells differentiating. Okay, so there's some ethical concerns about using embryos to carry out this sort of technology. So there's a couple of ethical considerations that you might want to think about and you might have to respond to. Okay, so we've got some safety concerns here. All right, so the possibility of off-target cleavages. So that Cas9 enzyme might accidentally snip another bit of DNA that we don't want to actually mutate in any way. Informed consent. Okay, so this is the problem with the embryo use. There's a lot of ethical problems with or considerations that we need to think about with using embryos and whether we think that that is an ethical thing to do and if the payoff is worth this process. There's also inequality. So when this actually does come out and is available for humans, this is going to be a very, very expensive technology to use. So will everyone be able to use it, or will only be wealthy people that can use it? And then, of course, discrimination moving on from that is that CRISPR may be a threat to those who are judged by society as biologically inferior, when in fact those individuals do not need fixing at all. So we might deem, for instance, a particular gene of interest to be detrimental to survival and we want to wipe it out, but some people might think, no, actually, I don't really want to wipe this out. I don't think it's detrimental to me at all. So there's a whole range of social and economical and ethical considerations that we need to think of when we're thinking about CRISPR. But I think the main takeaway is that we're not using this in humans yet, and these are a couple of the things that we need to think about before we do start to use it in humans. So thank you for watching. Please bring your questions to class as always, and I'll see you there.